I'm Lindsay Volk in Dr. Engelward's lab at MIT, and today I'd like to talk to you about my research. So did you know that every day we are bombarded by chemicals in our environment? Many factors influence how we respond to these chemicals, including the dose we are exposed to, as well as elements of our nature and nurture. Specifically, I study the involvement of an individual's DNA repair capacity, age, and sex in susceptibility to cancer-causing agents. Currently, I am investigating a chemical called N-nitrosodimethylamine, or for short, NDMA. NDMA is categorized as a probable human carcinogen and can be found in contaminated drinking water and medications, as well as in some of the foods we eat. Notably, one study revealed a link between NDMA exposure through drinking water and childhood cancer in Wilmington, Massachusetts. To really understand the effects of NDMA, who is more susceptible, and what we can do to counteract it, we have to dive into the mechanism. Like a recipe book, DNA holds the instructions for making all the proteins in our body. These proteins help maintain our physiology, and if the DNA code is altered, abnormal proteins can lead to abnormal physiology. Our DNA is constantly damaged from sources that are generated within our cells or that come from the environment, such as NDMA. If the damage is passed on to daughter cells in the form of mutations, cancer can arise. This is why our cells have an essential defense mechanism, DNA repair. Whenever our DNA is damaged, DNA repair proteins are quickly recruited and will either remove the damage or, in extreme situations, signal for the cell to die. It is better for the cell to die than for it to pass on mutations. Results from our studies have shown that DNA repair proteins 0,6-methylguanine DNA methyltransferase and 3-alkyladenine DNA glycosylase are essential in resolving NDMA-induced DNA lesions and preventing cancer in mice. These findings indicate that individuals with reduced DNA repair capacity may be more susceptible to methylating agents such as NDMA. Additionally, recent findings demonstrate a difference in vulnerability to NDMA based on age. This may be due to changes in the rate of DNA replication between younger and older mice. Lastly, we have observed sex differences, with male mice being more sensitive to NDMA. Further studies will investigate how DNA repair, age, and sex alter NDMA-induced carcinogenesis in animal models. Findings from this project may help to identify vulnerable populations and lead to the development of novel interventions.